female, and of course this is all about the gonads producing gametes for reproduction. So looking at just the anatomy right now. We have the gonad, which is the testis, enclosed in the sapien, the scrotum, the sac, and coming off the, on the superior aspect of the testis and the dorsal aspect here, you have the epididymis. I'm just going to show you the anatomy, and then we're going to go through it after the sperm is produced. All right, so testis here, and then the epididymis, this is within the scrotum. And then we have the ducts. All right, so we have the ductus deferens, or the vas deferens, same thing. And then here is the ampulla of the vas deferens. It's just a little bulge here, before it enters the ejaculatory duct. Now, also entering into the ejaculatory duct is the seminal vesicle. That's here. There's quite a bit of, ancillary structures contributing to the semen. So the seminal vesicle is one of them. So seminal vesicle joins where the ampulla of the vas deferens is, and this is now the ejaculatory duct. This is entering the prostate gland here. So here's your urinary bladder. This is the prostate gland. Coming from the urinary bladder now is the urethra. And the urethra will be named according to where it's traveling. So as it leaves the bladder and enters the prostate, it is the prostatic urethra. So right here is the prostatic urethra. The ejaculatory duct containing the semen is now entering the prostatic urethra. As the urethra passes through the urogenital diaphragm, the floor, the pelvic floor, then it's the membranous urethra. Right here, the membranous urethra. Then as it enters the penis itself, the copulatory organ, then is the penile or the spongy urethra. All right. And then, of course, the external urethral orifice here at the end of the penis. So that's the urethra. Also contributing to the semen, in addition, the prostate gland also contributes to the composition of the semen. The seminal vesicle and the bulbourethral gland here, that also contributes sort of a viscous fluid, bulbourethral gland. Okay, so those are the ducts, the ancillaries. The penis itself is going to consist of erectile columns of tissue. All right, so immediately surrounding the urethra, there is the corpus spongiosum. So all of this blue is representing erectile tissue. It's very vascular, connective tissue with a lot of vascular spaces, and very contractile, of course. So this is the spongy corpus spongiosum immediately around, and the spongiosum is going to start here at the root of the penis, and this is the bulb. The bulb is at the root. And then it continues distally to the glans penis. So this is all the corpus spongiosum, from the bulb to the glans penis. And the glans penis, of course, is covered by a skin that can, come back, and that's the prepuce, the skin that covers the glans penis. The other major contractile columns of the penis are the corpus cavernosum. And here on this mid-sagittal cut, you really only see this section of it, but really it's the majority of the penis. So this is the corpus cavernosum here. You see it. And I'll show you another picture of it where you see more of it.
That's the
So as far as the columns go. Okay, so again, here's the bladder, the prostate gland, bulbourethral glands, and the penis itself, frontal view. So you see that this is the penile urethra here, covered or enclosed by the spongiosum, right, from the bulb to the glans penis. And so this is it on the cross section, you see. So it really does take up a minor portion of the body. And then here, these are the corpus cavernosum, the two other columns. At the base they are, or at the root they are the crus or crura. This is the crura at the root. And then they can continue the length of the penis. Right? So these are the two corpus cavernosa in the cross section. This is the relationship now of the testis. This is now the testis and the scrotum in a bit more detail. So you have the skin of the scrotum, and then you have immediately deep to the skin, you have a muscle, the dartos muscle. All right, so this is the dartos muscle here, just deep to the skin. Then you have the testis that is enclosed in some capsules, tunics. So going from superficial to deep, you have this fascia, right, that's making up, this is the external fascia here, which is going to enclose all of this unit here. And that is actually going to be called the spermatic cord. So as the testes descend through the inguinal canal, before birth, it pulls with it all of this, and this is all called the spermatic cord. And when you go deep to that external fascia, you see this muscle. This is the cremaster muscle. So the cremaster muscle is really the one that's going to raise and lower the testis. This is all about temperature control, because the sperm are produced in the testes, and they require a certain temperature, a little bit lower than body temperature. So the cremaster is to lower the testes in warm weather and raise it in cool weather so that you have the proper temperature for sperm production. The dartos plays a role in that also by condensing the space, contracting the space for the scrotal sac. All right, so this is the cremaster muscle. This is within the cord, the spermatic cord. When you go deeper still now, you see the actual tunics around the testis. This is the vaginalis, the tunica vaginalis, which has two layers, and that's the external tunic of the testis. Then the internal tunic is the albuginea, and that's what alba is white. So the albuginea is the white tunic that's right on top of the testis, and you see it invaginates into you compartmentalize the testis itself. Here is your blood supply, right? The testicular artery, and the veins are called the campaniform plexus of the testicular veins. The reason here is this vein is sort of important is that it cools the arterial blood. Another way to regulate the temperature. So we have quite a bit. We've got muscles, and we have the veins cooling the temperature of the arteries. Okay. So looking at the testis now, that albuginea, the white, so this is vaginalis, the outer capsule. Vaginalis, and it's got a cavity, so it's a visceral, parietal visceral layer almost of the tunica vaginalis. The albuginea is right on top, and you see how it invaginates and compartmentalizes the testis into lobules. And within these lobules, this is where you find the seminiferous tubules. And this is the actual site of spermatogenesis, the seminiferous tubules. And then, so the sperm is going to be produced here in the seminiferous tubules, and then the sperm continue out through the epididymis, where the sperm are stored in the epididymis, and then they will be ejected out through the vas deferens. This is what the slide is going to look like. This is that first slide. And so if you take a cross-section here of these seminiferous tubules, you have all of these cross-sections. right? And what we'll look at now is all of these cells in here that are producing the sperm. And in the lumen is where the immature sperm are now traveling. And the cells in between the seminiferous tubules, the interstitial cells, they are called the interstitial cells of Leydig, Leydig, the interstitial cells of Leydig. They produce testosterone. So all of this activity that we're going to be looking at, spermatogenesis, the making of the sperm, certainly requires testosterone. So it's all here in the testes, in the seminiferous tubules. So if we look now at the wall, this is the wall of the seminiferous tubule. We're going to start out here at the periphery. So this is basically spermatogenesis. So here's the wall. And out here, this is called the basal compartment. And here you have the stem cells. And they are now, so now you've got new language, you've got to know these cells by their name. All right, these stem cells are called spermatogonia. That's the stem cells, spermatogonia. And they reproduce mitotically to give you a tremendous population of stem cells. So in the male, this is happening from birth till almost death, pretty much, the lifetime of the male. These stem cells are producing a population of stem cells. At puberty, now what happens is the stem cell is going to now divide into an A and a B cell, which is sort of a differentiation. The A cell is going to stay here, and the B cell now is going to start the process of spermatogenesis. All right, the B cell is going to leave the basal compartment and enter the adluminal compartment here, adluminal to the lumen, right? And that's going to become a primary spermatocyte. This is going to start meiosis. What is separating the basal compartment from the adluminal is this. This is a tight junction. And believe it or not, this whole green thing, all of this, one here, see its nucleus, and one there, there's a nucleus. There's two cells, two big green cells, and these green cells here, each one is called a sustentacular cell. Or a Sertoli cell. All right, same thing. Sustentacular or Sertoli. So here's one nucleus, and so the cell is, all of this is one cell. Here's the tight junction. That's one cell, okay? And then here's the other cell. These cells are critical to sperm production, spermatogenesis. The Sertoli cells, first off, this junction that they form, this tight junction, this is to protect the sperm that are being produced, because these cells, these stem cells are there at birth, like I said. After meiosis, the whole point of meiosis is to cross over and make new cells, genetically variation. So these would be new cells that the immune system hasn't seen yet. Okay? So if these sperm get out and the blood, the immune cells see these cells, they would attack them as foreign. Okay? So this is a blood testis barrier. The Sertoli cells, they produce a blood testis barrier to protect the sperm, otherwise they would be attacked. So that's one thing they do. And that you see how they guide the cells as they become, spermatozoa, and then they enter the lumen. So they guide, they protect, they provide the nutrients, they help the testosterone, which are, they have binding sites for testosterone to again help the spermatogenesis activity. So Sertoli cells are absolutely critical for the seminiferous tubule in spermatogenesis. Okay, so what we have to look at now is meiosis, because this is the formation of the sperm. And the whole idea of meiosis is to produce a haploid gamete. That's what we're doing here. So just to remind you how this works. We start off, as adults, we have diploid cells. Except for our eggs and sperm, all of our cells are diploids. And diploid means that we have 23 chromosomes from our mother, let's say, and 23 chromosomes from our father. All right, when we became fertilized eggs, that's what happened. And then these, the 23 of each from egg and sperm come together, and that makes up the 46, the diploid number. And all of our cells then, diploid, continue to divide mitotically until you become you. When you want to start now making eggs and sperm, today we're going to do sperm, in the spermatogonia, they are first going to undergo, before you can, this is now at puberty with a male. If you remember this, remember that? The cell cycle? Remember what the S stood for? Way back, September.
Oh, no.
What happens in the S cycle, of the cell cycle? The S, this whole thing is interphase. But before it goes into mitosis, what has to happen? DNA has to double, right? During the S is when DNA replicates, you get a copy. Because the cell is going to divide into two, so you need two copies. So what happens here now, you have 46 chromosomes. And the chromosomes can be considered homologous pairs. So this is pair number 1 from the father, pair number 1 from the mother, let's say. They're called homologous pairs because they have the same information on them. The genes that are on this chromosome are the same information, like the gene for eye color, the gene for a certain protein. All of your information is the same on pair number 1. So from the chromosome you got from your mother, the chromosome you got from your father, that pair number 1 is considered a homologous pair. We have 23 pairs, right? That makes up the 46. During the S, if you're going to now divide, you have to first make copies, and that's replication. So what you see here is that every one of these chromosomes made a copy of itself. Right? And the copies now are called chromatids. Bring back, right? The copies are called chromatids, but they are considered a chromosome, even though it's 2 copies, because it still has the 1 centromere, right? So this is, 2 copies, here's 2 copies, but they're considered 2 chromosomes because it's just 2 centromeres that are connecting the 2 copies. Okay. So basically the objective here now is to, in meiosis, we want to get down to haploid gametes. We want haploid, we only want 23, because 23 of the sperm is going to join with 23 of the egg and get back up to 46. So that's what we have to do now. We've got to go through meiosis. And meiosis is going to have 2, it's like a reduction division. It's not just one series of stages like with mitosis. We have to go through it twice. So just to remember, interphase, is when everything is happening, like here, and we're making copies. So now we look like that. And when you set up, when the homologous pairs come together, this is really four copies, right? 1, 2, 3, 4. So there's 4 copies that of the homologous pair, and that's called a tetrad. So prophase 1, and you use the number 1 because we're going to have to do this twice. In prophase 1, this is the most important part of meiosis. Right? Meiosis is forming eggs and sperm. In prophase 1, when the tetrad forms, the really critical thing here is that we have crossing over. And the crossing over means that there's enzymatic breakdown, basically mixing and matching. The genes are going to break off. A gene from here will break off and join this one, so that, and vice versa. And genes from this chromosome break off and join on that one. So the result here, after crossing over, is that this chromosome is going to have information, genes or traits from the other source, the chromosome that you got from the other parent. This is to give variation, survival of the species. You want variation, you don't want clones, you want variation is good for the species. Okay, so this is the most important part of meiosis as far as providing variation. It happens in prophase 1. Then the rest of it's going to look very similar. The tetrad is actually now lined up on the equator, that spindle fiber that's formed. Now in anaphase 1, we do not split the centromeres, right? But instead, each member of the homologous pair separates. So it's just the whole pair. So this goes here, and this goes here. Right? We don't split centromeres. It's just the whole pair that separates from one another. So the result here is that from the primary spermatocyte to the secondary spermatocytes, so primary is 46. That equals the primary spermatocyte. Now when we get after anaphase 1, and we have 2 secondary spermatocytes, they're going to be haploid. So 23 chromosomes is haploid, and that's what we're going to have here after the first division. But the 23 chromosomes still have 2 copies attached. So this is not viable. You don't want to have this going into a sperm, because what would happen if that joined with an egg? If this has 2 copies still attached, and then it joined the egg's chromosome, you would have 3 copies of that pair. That's trisomy trouble, right? Trisomy 21 is Downs. I mean, some trisomies are really, much more problems. In any event, it's not the optimum way. You should have just 2 copies. Okay, so then we have to go through this again. This is going to look like mitosis more than anything else. Now the 2 secondary spermatocytes are now going to go through the same meiotic division. Nothing special happens in prophase 2. It's just the typical, the condensation of the chromosomes. In metaphase, they line up. Anaphase here, anaphase 2, is when now they split. All right, this is when the centromere splits, and now you have full chromosomes. There are no more copies. And that's how you end up with 4, because you start with 2, each one gives rise to 2. And that's how you have 4 haploid daughter cells. And these haploids now are single copies, and that's what you want in a gamete. You want 23 chromosomes, single copy. And with meiosis with the male, what's also significant is that the four gametes look similar phenotypically, not genetically, because it's crossing over, so the genes have some variation. But when you see it, the size is the same, the cytoplasm, they look similar. All right, so that's different, because the female, of course, is different than that. So if we translate that into the testis where we were, all right, the spermatogonia is the stem cell up here. After puberty, it becomes a primary spermatocyte. Primary spermatocyte now, after the first meiotic division, becomes two secondary spermatocytes. They are now haploid, but they have two copies. So each of those undergoes the second meiotic division, and now you have four spermatids. The spermatids, look similar in size and shape. They have to undergo maturation. They have to undergo spermiogenesis. Right? Spermiogenesis now is where you start to look like a sperm. So this is spermiogenesis. This, and by the way, Sertoli cells also play a role in this, absorbing the excess cytoplasm. Okay, so here, spermatid, you get four of these. You keep the nucleus. That's, of course, what it's all about, maintaining the nucleus. But everything else gets streamlined. The Golgi apparatus, which is all of your enzymes, becomes an acrosome. Right? So it covers the nucleus. That's the acrosome, and it was your Golgi. You also have, so that's like the head, right? The nucleus with the acrosome. The mid piece is now pretty much mitochondria. Why? Why would we want to maintain a mitochondria? ATP, right? The energy, you got it, motility. This is our only flagella in us, humans only. Bacteria loaded, we only have one, this is it. And it is, a microtubule. And so here's the energy for it. So this is a classic structure function. We have a cell that has the, has nuclear information it's going to deliver. The acrosome has enzymes that will help it break down on the trip when it gets into the vagina and the uterus. And then the energy and the motility to get there. Right? Pure and simple structure function. So that's spermiogenesis, and that takes place, again, traveling as it's coming through the wall into the lumen of the seminiferous tubule, and they're still not functional. I mean, they're considered spermatozoa, immature sperm. They don't have the motility yet. They'll get that pretty much as they travel through the lumen, and especially in the epididymis. That's where they really become more motile and functional, because they get stored in the epididymis, and then on ejaculation, that's when the vas deferens is going to contract and expel them. So this is a great picture. This is a real picture of a seminiferous tubule. The spermatogonia would be out here in the periphery. All of these cells represent cells of spermatogenesis, including Sertoli. So that's the wall. This is the lumen with all the sperm traveling. So that's one, in fact, that's one of the slides we have to look at. Okay, so that's the anatomy. And this is sort of physiology too, the meiosis. I'm giving it to you because I have models for it.